All right, thanks a lot. Let's see if I can make this work. All right, here's the pointer. So take note of that. That's the website for this project, and any of the information that you might want to have about some of the things that you hear today, including slide sets and things like that, contact information for the participants, and the names of the participants, you can find at that website. So go to that website, it will tell you how to track us down, how to get in touch with us, how to get specific information about the project from us, okay? So that may be the most important thing that I have to say right here today. www.beefefficiency.org. Okay, here are the participants. And one of the things I did notice, in fact, there's a couple of things I just noticed about this, is that my slide of participants differs a little bit from the one that Matt Spangler just showed you. And I show 18 investigators from 11 institutions. He has 20 from 10. I'm a little worried about the management of this project, that people sort of slipping in and slipping out that I don't know about. And so I'm not exactly sure what's going on here. But another thing you might notice down here is basically an indicator of things that you can do with your DNA. So if you're interested in genotyping your cells, you can find out things about your origin. One of the things I found out about my origin is that my mother's lineage come from somewhere right up in northern Russia. Who knew, right? I originated in the United Kingdom. I was born in England. I immigrated to Australia when I was about 4 years old. Grew up and went to school there and then came to the US, but never would have thought that my mum's family came from somewhere up in northern Russia. On the other hand, my dad's pretty damn common. He has the most common Y chromosome that's found in Europe, which is okay. I'm okay with that. I have a friend in Missouri that lives in Iberia who is like me. He looks like me. He's a little skinnier and fitter than me. But he has a Y chromosome that's the most common chromosome found in Bantu tribesmen in Africa. So you find out some interesting things about your family when you start doing this. One of the things that I found out was that 2.7% of my DNA appears to come from Neanderthals. My wife was not in the least bit concerned about that. She said she knew it all along. Some of you may know Dorian Garrick from Iowa State, and he's also a major part of this grant, and another geneticist. And Dorian checks in at about 2.9%, so there's no surprise there, I say. Okay, so this grant is all about trying to do a number of different things, but one of the major genetics foci, and I'm going to try to focus on that, because you've already heard a lot about some of the nutritional work that's going on. I'm going to try to focus on some of the genetic work that's going on in here, is all about trying to do something called genomic selection. Have any of you heard of that term, genomic selection? Hands up if you've heard of that term. About 2 of you, okay. We've been talking a lot about it, but that's a lot of the speakers, Matt and Bob, have been talking about genomic selection. They've just not used that terminology. What genomic selection is all about is taking a whole group of animals that you've got an important, valuable phenotype on. Let's say something like feed efficiency, feed intake, right? Because it's economically important. We do not routinely collect it within the industry, and yet we know that it's responsible for a huge amount of variation in the cost of producing our product and the profitability that people have at various points within the production chain. So if you can gather a lot of individuals, you can phenotype those individuals, get their dry matter intakes, get their average daily gains, get their metabolic midweights, get their carcass data, and then genotype them with some sort of assay. We're now in a position that we can do something complicated statistically. In other words, try to estimate the contribution that each one of those SNPs has to the genetic merit of the animal. And if we can do that, it's now possible to construct what we call a molecular breeding value or a genomic enhanced EPD that will allow us to actually get an estimate of genetic merit, an EPD on an animal based only on its DNA. So you take a DNA sample, you genotype that DNA sample, and you take all of the genotypes you get. There's maybe 50,000 of them, there's maybe 800,000 of them, and as you'll see later in this talk, there's going to ultimately be something in the vicinity of 25 million of them, because we're now at the point that we're starting to sequence whole genomes on animals. And what we want to do is be able to find the ones that contribute to variation in traits, estimate the value of being an AA versus an AB or a BB at that locus, and say, if you're an AA, I add 1 point to your EPD. If you're AB, I add nothing, and if you're BB, I take a point away. And then I go to the next marker and I do the same thing. And the bottom line is, you're doing what you guys would have come across in estimating genetic merits, EBVs, when you took an animal breeding class 40 years ago. The only difference is we're now at the point where we can get a higher resolution look at the genome of animals. And so that's a really big part of this grant. To do that, when we proposed this grant, we said we would collect DNA samples and phenotypes on 8,000 animals representing 8 of the major breeds. And this basically gives the breakdown by year of the project. We are right here in the project right now. So if we were on track, we should have produced about 5,000 animals. And this shows the breakdown by the individual breeds, where we have both intake data, carcass data and the like, and in some cases genotypes, not in all cases. And you'll see that right out here is where Charolais really start to kick up, with the 450 animals in each of those production groups that are being produced here right now. So I'm going to show you some analysis of data, but not for this particular group, not your group, because we don't have genotype. We don't have all of the phenotypes yet, but they're coming. Bottom line is about 7,000, 8,000 animals representing about 8 different breed types were what we proposed to be able to do. We had money in the grant to actually genotype only about 2,400 of them. And the problem is, we say this is a $5 million grant, and that sounds like an awful lot of money, and it is an awful lot of money. But when you genotype 2,400 animals and you use an assay that costs about $200 per animal, you do the math, you add the indirect on top of that, and 20% of your budget's going straight away just to get these animals genotyped. But fortunately, because at the University of Missouri we'd been working in this area, and at Illinois you had been working in this area, there was legacy data floating around. So we had 700 Angus calves that were fed at the Circle A ranch that we have phenotypes, and we'd already genotyped. And there was a couple of thousand, 1,400 animals, I think, that came from the University of Illinois that were Simmental Angus cross animals that had been fed here, been genotyped on another project. So we started this project with 2,000 animals that were already genotyped in the can. So we said we'd do another 2,400, which would bring us up to about 4,400. As you'll see, we're doing a whole lot better than that for a whole number of reasons. We were going to use these data for a lot of different reasons, most important to build these models, to predict EPDs using phenotypes and genotypes. Because if we can do that, we can then move towards the commercialization of that technology within the individual breed associations. We want to do a whole bunch of other things and try to dissect out where the genes are that really influence variation in feed intake and feed efficiency. So we don't just want to build these really generic models that say, you know, here's 800,000 markers, let's go down that list for an animal, add a point, take a point, add half a point, take half a point. What we really want to do is go in there and identify the individual genes that cause variation in those traits. Because if we can do that, there's lots of advantages. Number one, it'll work across breeds. And number two, we don't have to genotype 800,000 SNPs for $200 every time we want to do a test. That's too expensive. So, a number of different things that we're going to be used for that. We're also going to look at different parts of the genome, the mitochondrial genome in these animals, come in and try to identify where these genes are. Greenhouse gas work. There's also work that's being done at Clay Center by Harvey Fruitley, who has a facility that looks very much like the facility that you saw here today, where he's measuring greenhouse gas outputs from animals of known feed efficiency, and trying to look at, are efficient animals higher or lower in greenhouse gas emissions than inefficient animals? This is going to become increasingly important. If any of you guys ever go to Europe and go to meetings in Europe that involve producers and start talking about what are the most important issues that they're facing over there, you're going to find things like greenhouse gases. So I've been over there and given talks 2 or 3 times in the last couple of years. 2 years ago, I talked at the Breeders' Convention, the British Breeders' convention in England, and I said, what are you investing all of your money in terms of genetic improvement? They said greenhouse gases. It was the most important thing on their agenda. And the reason is, of course, that people are telling the politicians that 60% of all of the greenhouse gases that are influencing our climate change are coming from production agriculture. And what do you do if you don't measure it? Dan already said that. We've got to measure it, we've got to quantify that, and be able to respond to that. And actually probably deal with that from a genetic perspective or other management perspectives as time goes on. It's going to become an increasing issue for us. We don't want any of this stuff to go to waste, so we actually have a repository at the University of Missouri. We're going to duplicate this somewhere. So I told you that we've got 8,000 DNA samples. We're headed towards 8,000 DNA samples, all of the phenotypes, genotypes on all of these animals. Dan talked about how big a problem he was having putting a database together. Our database at the University of Missouri currently has 12 billion genotypes in it. The sequence data in that database is now currently about 100 times larger than that. We need 40 terabytes of data storage just to store this stuff. You don't want to lose all of that. It's important, it's valuable. The government's paid for it, you have paid for it. We need to make sure it doesn't get lost. So one of the things we're doing is actually giving as much of this data back to the breed associations as we possibly can. It's your data, you're going to make the most use of it. Let's push it back so that you can use it. We need to tell you how to use it and work with you to actually make it of value to you. But that's one of the ways that we're trying to make sure that these data don't get lost. Dan already told you about this experiment. We had money budgeted to genotype about 400 of those animals. My plan now is we'll genotype all of them. I think we're going to have the money to do that, but we're also, an important facet of this is sort of training students and training undergraduate and graduate students who are going to be the focus of the industry. That may not seem so important to people like Bob Weber and Dan Scheich and Matt Spangler, who are all young whippersnappers, but I'm getting to that point in my career that I know that I'm not going to be around doing this for more than probably another 8 or 9 years, and it's really important that we actually keep the flow of knowledge and trained individuals coming along behind us. So that's a big part of this grant. The extension objectives you've already heard, so I'm not going to go across there, other than to say we're already here in terms of where we're going to. The first 220 Rex Ranch animals were fed at Missouri. We've gathered the data on those, and they've been genotyped. I just got the genotypes back. We're ready to actually take the genotypic data, the intake data, push that back to Matt, push that back to the Rex Ranch. And in the future, we're going to be actually taking animals from that system, dropping them into feedlots, and actually predicting what their intake is going to be when those animals go through the feedlot. So the feedlots will validate for us on a group basis whether or not our predictions of intake and feed efficiency on these animals match up with what they see. And at that point, I think we're going to have something that'll be pretty valuable from the industry perspective. So kind of where are we at? That's what we said we were going to do, and I'm going to focus now on a couple of the things that we are doing. So where are we at? Well, where we're at is at the end of 2013 alone, I think we're going to be at 9,000 samples alone. We've been pretty busy whipping up animals and getting them genotyped. So already genotyped, these animals in this column with a high density assay, and in this column with a 50K assay. So you may or may not know anything about the sorts of genotyping technologies. The point is, this one does 50,000 SNPs, this one does 800,000 SNPs, this one is more than twice as expensive as this one, and in practicality, it doesn't seem to give us a whole lot more information. So what we actually start doing is every time we start genotyping individuals from a new breed, you'll see we've got 24 Charolais here already, we genotype them with the HD, we do that till we get 200 or 300 of them genotyped, and then we swap over to the low density assay where we're saving money and try to genotype as many animals as we can. So you'll see that there are several large groups here. Angus, of course, fairly large, Hereford's fairly large, Sim Angus, this is a really big legacy group that we have. And we've analyzed some of those data. This is the number of SNPs that we've got that were genotyped in these groups, and I'll show you some data from these analyses in just a minute. We are also in a position where I think we're going to be able to get a bunch more data from Ireland and from the Canadian Feed Efficiency Project. So the Irish Chuggas group have been doing feed intake on individual animals. They've indicated that they're interested in swapping data with us, making their data available to us. The Canadians are feeding a whole bunch of different animals, also about 8,000 animals. The breed compositions of those animals are a little strange. They're not as purebred as we have sort of represented here. Most of their animals are composites. We have some composites that are two-way or three-way crosses. They have animals that are multi-breed composites in their populations. So I'm not quite sure how that will compare with our data, but we're about to find that out, and then we'll swap data. So we could be at a point within the next year or so that we actually have something like 12,000 animals that are genotyped. And so by pooling data from different populations, I think we'll be able to get to the point that we have enough data for a number of the breeds, including Charolais, that we'll be able to get this technology running for many of the different breeds. So here's the data that we've looked at so far. 5,000 animals genotyped. They include some Hereford animals that were fed by Doug Olson. About 850 of them ended up in the analysis. F1 squared composites. These are the cycle 7 animals at the US Meat Animal Research Center. They make an F1 by crossing 2 breeds. They make another F1 by crossing 2 different breeds. They take those 2 different F1s, cross them, and basically make a 4-way, something that looks pretty much like a 4-way cross. And they fed 1,160 animals that ended up in our analysis. There are about 1,400 legacy Simmentals, Sim-Angus animals that were fed here at Illinois that were in the analysis. About 1,580 Angus animals that ended up in the analysis. And this is a little table. It's got a lot of information in it. But what I want to just draw your attention to, here's what the heritabilities are. Now, these heritabilities are based on an analysis that uses marker information, not pedigree information. We don't know the pedigree of these animals. Don't know the pedigree of nearly all of them. Some of them we do, but most of them we have no idea what the pedigree is. We establish their pedigrees or their relatedness using marker information. So we take their genotypes and we say, essentially, what is the correlation between your set of genotypes and your set of genotypes? And that tells us how closely related you are. Now, in this case, that correlation is probably close to 0. But in these populations, there are a lot of common sires used, common dams used, and so we can establish fairly accurately what the relatedness is. So the heritabilities we're seeing for dry matter intake across all these populations are really quite similar, 27% to 41%. This is metabolic mid-weight, so it's a weight trait, and as you'd expect, nearly everything's 50%. Here's growth from 19% through to about 30%. These are very typical from what you see from analyses of national beef cattle evaluation databases. And here's RFI from 21 through to about 45%. Now, what underlies the fact that we see differences here? Why do I see a heritability of 27% here versus 41% here? I think it's got to do with a number of different things. It's got to do with the feeding system, the number of contemporary groups that are represented in the data set, stuff like that. I also think it happens to do with the fact that these are crosses and these are not. And one of the interesting things I'm seeing is the heritabilities look as though they tend to be a little lower in the crossbred animals relative to the pureblood or near pureblood animals that are in this population. Good news is these heritabilities are all pretty big. They're in the moderate range. It's going to be possible for us to make genetic improvement for all of these traits. I'm really glad every other speaker put this set of equations up here because I was a little worried about getting to this set of equations. This set of equations, other than today, have you seen these equations before? So these up here, have you seen those before? So these are Henderson's mixed model equations. He came up with them in 1949 and he published them in this format in 1959. It took him that long to actually figure out the properties of all of this stuff. By 1971, these equations were first used for cattle evaluation, and Paul Miller at Cornell was the guy that first implemented this technology. And the compute power back then was so sophisticated that he could nearly get 500 animals into the evaluation system. And now today we do this with millions and millions of animals. There is one difference between Henderson's formulation in the formulation that I'm showing you here, and that is this matrix here. He used something called A. A is a matrix that is based upon pedigree information. It says how much of the genome would you expect two individuals to have identical by descent based upon their pedigree. And now what we're using here is a matrix that's based upon genotypes. And you asked a really good question earlier about EPDs computed based upon a phenotype system and computed based upon a genotype system. There are a couple of inherent incompatibilities between those two that we haven't quite figured out how to solve yet. And Bob Weber was talking about, well, we can blend them, or we can do this, or we can do that. But here's the problem. I work with a data set that is actually from Angus, where I have 3,570 Angus bulls that we've genotyped. These bulls were born between 1955 and 2007. And when I get the pedigrees for these animals and I track their pedigrees back, what we say is when you get an animal that you don't know who the mom and the dad were, right, the sire and the dam, they're missing, by definition we call them a base generation animal. And if you look at the pedigree of these 3,500 bulls and you track back their pedigrees until sire and dam information disappears, those animals are born between 1836 and 1940, 100 years. And when we do national cattle evaluation, we say, oh, they're all the base generation animals. So they're all that original progenitor population. Well, they're 100 years separated in time. The other problem with it is that because we don't know who they are, we say they're all outbred animals. There's no inbreeding in any of those base generation animals, so we put that in there as being 0. What that does is it calibrates our EPD system, right? And there's not much you can do about it, right? I can't go and get an animal born in 1836 unless I can dig him up and find his bones and identify who it is and take his DNA out and actually calibrate what I see in the genomes of the animals that I've genotyped relative to the pedigree information. When I genotype this animal, I don't need pedigree information. I just ask the question, how related are they based upon their DNA? I ask the question, how homozygous are they based upon their DNA? And that tells me how inbred they are. So there's an incompatibility that's built into this that we haven't quite figured out how to solve yet. But from this perspective, it's not going to be a problem because you're not going to have EPDs for feed efficiency based on a whole bunch of animals that have pedigree information unless you sort of try to merge those. If you have animals that you have feed intake and no DNA typing on, it becomes a problem, right? So, from my perspective, this is valuable because if you genotype young animals at birth, the question is, can you generate something that's more accurate than a mid-parent, right? Just a mid-parent estimate. And we published 2 papers last year where we analyzed these 3,500 Angus bulls. We analyzed about 2,400 Simmental bulls and about 2,700 Limousin bulls. And what we show is that we can generate accuracies based upon DNA for all of the traits in national cattle evaluation that range between 60 and 80%. This technology works. It works. If you genotype animals at birth, use that information. If you gather phenotypic data on them in time, go to the national cattle evaluation, but you want to use this information at the point that you are making selection decisions on young bulls. What we do is we crank the handle on this, we then do this. That's another sort of little statistical trick where we take the molecular breeding values for these animals, we estimate SNP effects. Once we've got those SNP effects, we can genotype animals, put them in M, and estimate their additive genetic merit. Nice little mathematical way of saying how we go about implementing this technology. Okay, this is the real mathematical equation. Turns out this is equivalent to doing this, and this is something that Henderson published again in 1974. Henderson was a brilliant guy. I shared an office with him for about 2 years when I was at Cornell, and people used to say God spoke to Henderson because he would see this stuff and he would just write it down on a piece of paper, and then he'd come and start talking to people and say, can you help me prove this? So people like Dick Quast that were there and Charles Searle that were there would help him prove all of this stuff, but he just saw it. It just came to him in his dreams. This is a modification of the equations that we saw last time. And what this is here's the animals that we have data on in that set of equations. Here's the animals that we want to estimate their genetic merit. We don't have any data on them, but we can, we can, yeah, blew that, didn't I? So we can genotype them, and we can figure out how related they are to the animals that already have data. And so that's exactly how this technology works. If you have animals that are very closely related, that's the relationships that go in there. So these are the animals that you want to get estimates of their genetic merit for feed intake, right? They're young calves, they're on the ground right now. I genotype them, I figure out how related they are to the animals that are in the pens over there that we've just fed, and we've got DNA on and phenotypes on, and I estimate their genetic merit. If that's 0, this is 0 and there's no accuracy. If they're unrelated, there is no accuracy. If they're sire-son pairs, you've got the highest possible accuracy that you can have using this technology. Now I want you to remember that equation down there because there'll be a pop quiz at the end. I'm going to ask you to tell me what that equation's all about, okay? So that's why this technology doesn't work if we build our prediction equations up here in Charolais and then we run over and genotype a bunch of Angus and try to predict what their feed intake's going to be. The reason for that is, if you think about an average Angus animal and an average Charolais animal, and you track their pedigree back, how far do you have to go till you find a common ancestor? Hell of a time, right? You know, probably, I would say probably 3,000 years is how far you've got to go before you found the common ancestor to all the animals that ended up going down into France versus the animals that came later through the northern part of Europe, crossed over into Scotland and got down into Scotland. There were 2 separate invasions of cattle that did that. And their ancestors are long, long gone. So the coefficient of relationship between those animals is 0. That's why this doesn't work. Now that's bad news. It's bad news because it means that we can't develop this technology in Angus and then just give it to everybody, you know? Because we had a bunch of Angus that were genotyped, we had a bunch of Sim Angus that were genotyped. We can't just use that and say, Charolais, here's your prediction equations for genetic merit for feed intake in Charolais cattle. It doesn't work. They're not related to each other. It doesn't work. And it also means this. It also means that we're feeding a whole bunch of Charolais cross with Simmental Angus animals right now, and we're going to generate prediction equations which we want to use to predict genetic merit for feed intake for you guys, and the first crop that we do that will be the maximum accuracy you ever see. As time goes on, if we do not keep capturing feed intake data on these animals and retraining as generations advance, the accuracies are going to decrease. Now, they're not going to go to 0 overnight, but ultimately they're going to decrease in time. So it really means a couple of things here. It means that in the future we do need to be thinking about capturing DNA samples and phenotypes on every animal that we possibly can so that we can retrain. And it also means something that I'm interested in doing. We need to find the genes. We just need to go in and find the genes. And here's some of the signatures of those genes. You saw a couple of these earlier. These things are called Manhattan plots. Each one of these is a chromosome. So it goes from 0 up to 29. Different color means different chromosome. Every dot is a marker, a point on that chromosome. And every point that goes up here, height-wise, tells you how big an effect it has on that trait. And what you notice is that, okay, here's dry matter intake in Angus, here's RFI in Angus, here it is in Herefords. What you notice is that there are relatively few dots that go above those lines, but a couple of them seem to be pretty important. This one here explains 10% of the variation in dry matter intake in Angus. This one is 4% in Herefords. The weird thing is, this one doesn't look like it's here, and this one doesn't look like it's here. We're getting very different signatures of where the genes are that influence these traits in different breeds of cattle. And this shows another way of looking at this. So this is the Angus animals, the Hereford animals, the Simcross animals, the F1 by F1 cross animals from US Mart. This is dry matter intake. This is metabolic mid-test weight. This is gain on test. This is residual feed intake. And these are like the top 10 positions in the genome that we find a signature of a gene that influences these traits. What you notice is that there's a couple of things that I got in boxes here. This is on chromosome 20, at about 4 million bases on chromosome 20. There's something that pops up in Hereford, in Simmental, and in these crosses, but we don't see it in Angus. The same with chromosome 7 at 93 million bases. There is a gene that appears to influence gain, metabolic midweight, and even residual food intake in 3 of these breeds, but not in Angus. Interestingly enough, there's nothing in common between any 4 of these breeds for dry matter intake. And we don't understand what that means. We don't understand whether it means that there are just different genes that control these traits in the different breeds, or we're not finding them because of the assay that we're using, characteristics of the assay that we're using. And we won't know the answer to that probably until about a year from now, when we get bulls sequenced from all of these breeds, and we will know how similar and how different they really are at the level of the DNA. So, we've got some ideas about how to identify those causal variants, and one of the things that we're capitalizing on is a project that we've started at Missouri, where we're actually entirely sequencing the genome of 150 bulls from 7 different breeds to find out what the hell's in there. But most importantly for this particular project, what we're trying to find is genes that are broken. You might call them genetic defects, but when you get 2 copies of them together, they're lethal. So if you get 2 copies of a mutation together that causes curly calf, that's a genetic defect. If you get 2 copies of a mutation together that causes embryos to fail to develop, fail to implant, you get a lethal. What we've got is some data that we've got already that suggests there are a lot of these within breeds. We've got evidence of at least 170 of them within 11 Angus bulls that we've genotyped already. How do we know they're lethals? We know they're lethals because if you knock them out in mice, they're lethals. So we know from existing data that they're lethals. So when we get these bulls genotypes sequenced, and we already have 40 bulls sequenced, we have a total of 74 in the pipeline at the moment, we'll know a lot of these answers about what the variation is that really exists within these different breeds. We're also looking at gene expression. We want to know if we take different tissues on these animals, how do the genes get expressed differently? Could that be responsible for differences in feed intake? And if we find genes that are differentially expressed and we go across to that previous SNP analysis that I showed you, do they coincide? Because then that'll point, those SNPs will then point to the genes that we know are differentially expressed that may underlie differences in feed efficiency. So we've collected a lot of tissues. This actually, I forgot to put the last 12 animals that Dan just reminded me of about 2 days ago. We collected another 12 animals that are your animals, Charolais, the Charolais sire animals, 6 high, 6 low feed efficiency. That was done back in the spring. They're dead. We've got tissue samples. And these are the tissues that we collect out of all of these animals. And probably the most important ones to look at are hypothalamus in the brain, which regulates appetite, and probably liver, which is the energy source inside most mammals. So we've actually sent a whole bunch of DNA, a whole bunch of samples off to Scott Fahrenkrug at Minnesota to actually do the gene expression work. So that's already happening. We've already done some work in this area using animals that came from the Circle A Ranch, and so we have about another 38 animals that we've collected tissues from, and we've actually done a little bit of analysis on those already, and we're getting ready to publish some of that stuff. So we've got a lot of sort of information on how genes work in animals that have high efficiency versus low efficiency, and in conjunction with the SNP analyses that we've done, I think it's going to help us point to the genes that are important. This is a really cool figure. I just wanted to show it to you because this shows 8 animals, 4 that are high feed efficiency, 4 that are low feed efficiency, and this is their entire genome. So this is the 25,000 genes that John talked about that indicated, and the little bars here show you where the genes are highly expressed or lowly expressed in each animal, each of the 4 animals. And the resolution is not great, but there are regions in here where we can actually find genes that are lowly expressed in the low feed efficiency animals and highly expressed in the high feed efficiency animals. So it really is pointing, I think, that there are genes that work in liver that have effects on the feed efficiency of these animals. Finish up by a couple of other things. Let's talk a little bit about methane. We've talked a bit about this today. This is Harvey Freetley's work. 2 experiments done at Play Center where out of 2 groups of steers and heifers, reasonably large contemporary groups fed together, they identified 37 steers and 46 heifers. The little black dots are the ones that they selected, and this plot shows dry matter intake against basically growth. So they took the animals that were deviated most from the average, but right across that spectrum, and they said, let's measure methane emissions on these animals and see how the high efficiency versus low efficiency animals perform in terms of their methane emissions. And here's what they found. Now the bad news is the slopes of both of these lines go in the wrong direction. This is methane production measured on a per unit of dry matter intake basis. So for each pound of dry matter intake, how much methane did we see these animals produce? This is RFI, and remember, what is the RFI that we want? Positive or negative? What's good? Negative. So the negative RFI animals are more efficient, and what he found in both of these experiments was that the more efficient animals produced more methane from the head based on a per unit of dry matter basis. But what's different about efficient animals and inefficient animals? They don't have to eat as much, right? Per unit of growth and maintenance, they don't eat as much. And so what we haven't done, and what I haven't seen him done, is actually say, all right, now let's take this data, let's take these groups of animals, and for the total amount of period that they were fed, and the per pounds of beef harvested, what was the total greenhouse gas emission? So we need to do that to get a true handle on greenhouse gas output on an efficiency of beef production basis. Okay, we do have the repositories. Our labs look like this a lot of the time if you go to the freezer trying to find samples. We also have 30,000 DNA samples. We have tissue samples all over. We have 6 different freezers in 3 different rooms in the building. Fortunately, we have a very good database. So if I want to find a liver sample on one of the animals that was slaughtered here, I go to my database, I look up that animal, it tells me exactly where it is in the freezer. We want to share this information with people. So we're already talking about sharing phenotypes and genotypes with different groups. We're sharing information with the breed associations as we gather it. We want the information to be used. And that concludes what I have to say today. So I know that I'm standing in front of us with a beard, so thanks for your attention, I appreciate it.
Thank you, Dr. Taylor. Anybody have any quick questions for Dr. Taylor or any of the other speakers? I think we've still got all the speakers here if you've got any other questions.
I do have a question. I don't want to deviate too much, but something Jerry said made me think about this. Just in the last few weeks, there's been the announcement by Simmons-Hall and previously by Hereford that during their recalibration they had some issues. Could you talk briefly about that and what those issues were?
Yeah, I'm not the one to talk to. Dorian is the one to talk to on that. He's the guy that's actually physically working with them to actually translate the technology. But I know what causes it. It's what I actually mentioned. It's the fact that there's this single step approach to computing EBVs or EPDs that takes phenotypic data on the animals that you've got it on. It takes genotypic data on the animals you've genotyped, and it takes pedigree information on the animals that don't have genotype information. And it does an evaluation, so it's kind of the blending approach that Bob was talking about, except it's not blending. It's trying to do it all at once. It puts the relationship matrix in there, it puts the genomic relationship matrix in there, you crank the handle, you get estimates of genetic merit, and it's supposed to be all over and done with. Problem is it's not. And the reason it's not is that the genomic relationship matrix is not calibrated relative to the pedigree relationship matrix. Now, what do I mean by that? What do I mean by calibration? Well, calibration comes from two different things. It comes from how far back your pedigree information goes, and whether or not you've got inbreeding coefficients, errors in the pedigrees, all that kind of stuff, on the pedigree relationship matrix side. But it is calibrated relative to this thing that we call the base population. In other words, the group of animals at which everything goes to zero, all pedigree goes to zero. Now, you may slide your base around, but all that is an artificial recalibration, right? If you have a moving genetic base that you change every 5 years or so. Otherwise, your base generation is zero, everything is relative to those animals. When you genotype animals, that genomic relationship matrix actually depends upon allele frequencies. So the frequencies of each of these variants in the population. And you can only compute that for the earliest born animals that you genotype. So in Angus, these 3,500 animals that I genotype, the earliest I can go back and compute allele frequencies is for the period about 1955 to 1970. So I can get a slice of allele frequencies. And you may look at me and say, why don't you use all the genotypes to get allele frequencies? Well, it turns out these populations, your cattle populations, are under selection, and allele frequencies are changing in time. We published a study about a year ago now that looked at that in Angus. There are alleles in Angus that are moving so fast that they're completely changing from the A allele to the B allele over about a 50-year period. So the strength of selection that you guys are applying to the genome is not minuscule. It's not like allele frequencies just bouncing around like that. 80% of the loci in the genome are changing like that at amazing rates. So when I say calibration, so I've genotyped animals here, right? From here to here, and I know what allele frequencies are right there. The pedigree relationship matrix is trying to drag that back to a point in time that has another 40 or 50 years of selection that's gone on. So they're out of sync. These two matrices are out of sync, and we haven't figured out quite how to get them synced. So that's the issue. So people are now struggling trying to look at ways as to how to synchronize those two to be able to make sure that the animals that come out with genomic estimates of breeding value don't get artificially inflated, which is what's happening. So they're coming out with higher estimates of genetic merit than they really should, because those things are out of sync.
Thanks.
Any other questions for any of the speakers? Yeah, we're going to put this information we've been filming, and so we'll have that up on our web in maybe a couple of weeks. We'll have some links to those. Come back and watch it. However, also I think all of it as well as the PowerPoint presentations. A little different version will also be available, so we'll make that information available to you. And we've also got someone to write us some summaries as well. So we'll have a lot of information. Follow up, behind this today's conference here. Now tomorrow morning we'll meet back at 8 o'clock. I'm not sure if it's this room or a different room. So we'll have our workshop tomorrow morning at 8 o'clock with our Breed Improvement Committee, our Commercial Committee. So all of you are welcome to attend that. We'll also have, I think, Danny may want to speak to RFD-TV tomorrow. We'll be doing some filming, I think, out at the farm maybe, and also be interviewing, possibly some of us here as well. Interviewing some of us. So we'll have that tomorrow, and then we'll wrap up tomorrow at about 12 o'clock. Neil, you have anything else to say before we break the sitting?
Just thank our speakers.
Yep. All right, let's give our speakers a round of applause.